Okay, take your seat. We shall start. So before I jump into the topic, can you hear me well? Like, not too well, like, because I get an echo. Anyway, can you tune down my volume? I feel like a rock star now. Can I get more on the guitar? Anyway, so next week we have a midterm. This is the last lecture that's going to be on next week's midterm. That doesn't mean you can just skip next week's lectures because they will be covered in the following midterm. And we're going to have a review session on Wednesday between 6 and 8. So where both Carlos and I are going to come, probably sequentially for about 1 hour each. So that's one chance to have like a discussion. We can go over some of the material. The couple more announcements. Recordings. The media streaming is not my friend. So my recordings can be found under the resource. Did you guys find them? Some of them I could put them on the media stream, but I struggle with that. So I just record the file and put it under the resource folders with the lectures. Right? You got that? And last point, I've posted homeworks. Those are meant for you to help as like study help. Right? I don't need to see that, etc. So you have keys in separate files. My recommendation is you use them and try to retrieve the information, right? So you study, and I'm going to say that again, you pay attention, you're at it, you're actively studying, you're going over it again and again, and the fourth point is you test yourself on the material. Right? You just don't reread. You try to retrieve these memories, and there's all kinds of neuroscience behind this logic. Okay. With no further ado, let's jump into this fourth lecture on cell compartments. We have many of them. We're going to focus on endocytosis and targeting proteins to the lysosome. So indeed, and I'm going to again shamelessly walk you through the stuff we've talked about many times for the purpose of making, driving it home for real. So what happens here? Co-translational translocation. That's right. Right? So a signal peptide or a signal anchor triggers recruitment of SRP, pauses translation, brings the whole complex to the membrane, interacts with the translocon, starts translocation, SRP leaves, and the fact that SRP leaves helps translation resume. Okay? So then the protein is being put into the ER, and what happens at the same time? The previous lecture. Modifications. That's right. What kinds of modifications? Glycosylation, that's right, and glycosylation, and say it again? The sulfate bonds, very good, and the folding and so on. Great, right? So now you guys really are getting the picture. Now the proteins are ready, let's say. So how does the system know the proteins are ready? In the ER. Not quite. Yeah, so it has to do with the modifications of the glycidic residues, right? So those glycidic residues, they help the quality control. And removing residues one at a time helps, passing the protein along is like checking boxes for the system to say, well, that protein, is good to go, okay? And then eventually, once the protein is fully, licensed to move on, what happens? It just go back home? No. It gets packaged into a vesicle, right? So what's the trigger? Give it to me. A small GTPase, SAR1, right? SAR1, it gets activated. And again, the GTP-bound form is the active form. That's true for all the small GTPases we're going to talk about. That helps recruit the first components of the code. Right? The code. So the COP2 codes help form the vesicle, package it nicely, not forgetting what, because indeed, that's why it's called T-snare. In the vesicle, there are V-snares. So V-snares, and what else will help with the next step? The RABs, that's right. Right? So with the RABs and the V-snare, now those vesicles are ready to go and fuse with the target membrane, which is where? In the cis-Golgi. That's exactly right. Now, what happens in the cis-Golgi? We're doing the review session now, basically. All the further modifications, right? And we're going to see today that there are some tricks for some proteins to avoid that. All these sequential modifications. And then what happens in the Golgi apparatus here? It matures, right? That means it changes over time, right? So those changes involve the activity of these enzymes, which modify the sugars, and so on. And how is the system, so both the ER and the Golgi maintained? You know what? Retrograde transport. That's right. Now you guys are fully primed to succeed. That's very good. Okay, so today we're going to focus more. So this is all about anterograde transport, and we've already touched upon this retrograde transport. And we're going to really focus on what happens here, both in the very late stages of transport from the trans-Golgi network to places like the lysosome, but also endocytosis. We touched briefly upon on secretion. You can't do that to me. It's okay, I am a professional. I can handle that. So anyway, we're going to talk about endocytosis as well. So how is material now not just produced in the cell and exported, but taken from the outside and imported into the cell? And so everything we're going to talk about today happens at the trans-Golgi network, a lot of it, as well as at the plasma membrane. And one of the key players, I started previously talking to you about the different kinds of codes, right? And I already alluded to the idea that those different molecular identities, they convey information, right? So COP1 is specialized in retrograde transport, and COP2 was specialized in ER to cis-Golgi transport. Now, almost every other kinds of transport, vesicular transport, involves clathrin. So we're going to talk briefly about clathrin, which helps package vesicles both from the trans-Golgi network as well as from the endocytosis. Okay, never mind. So that's what we're talking about. Sorry, I'm going a bit ahead of my slides here. So let's just focus on clathrin. I've shown you this slide before, and again, I want to really show it again, shamelessly, to really impress upon a couple of the structure-function relationships that are really obvious and important with clathrin. So clathrins actually come in two different flavors as far as the original protein is concerned, the gene. So there are heavy chains and light chains, and this is what's depicted here. So the heavy chains together, they assemble three of them, and they form this so-called triskelion, and the light chain help keep the triskelion together. So the triskelion is already a higher order assembly. Okay, so I showed you this kind of hand model I showed you this the other time. So that would be one of the triskelion here in red, another one here in yellow, in green, and so on. And so the whole idea is that by further assembling this triskelion in a higher order molecular machine, you're basically creating the shape of the vesicle, right? And I'm going to show you this neat movie. Although it was pixelated for me last time I checked. Yeah. So this is a triskelion. And what you can really appreciate here is that in volume, you have these globular heads pointing inwards, right? And then the assembly of the different triskelion. So it's completely hardwired, right? There's no room for variability here. But eventually, the higher order structure now takes the shape of a vesicle. So you have, again, a direct relationship between the protein sequence, the structure of the different light chains and the triskelion, and the higher order business end of making a vesicle, right? And not just making a vesicle, but also interacting with the inside, right? So that's the whole idea behind these globular domains, which I showed you were pointing inwards, right? So what do you think are those good for? Yeah.
I wanted to ask, actually, if they always form vesicles of the same size, and how they determine the size between the angles.
That's a good question. Actually, what would be your guess?
Let's go around.
Actually, no, it's fairly constrained.
Okay.
Yeah, I mean, there might be some wiggle room you could imagine, but if you take this modeling, because of course that's a model, right? Just to be clear, this is not directly imaged from what happens. Well, it's fairly constrained, right? So it's really a molecular machine. It's not like a famous. So we may talk about that in the future, because there's an emerging theme. Sorry, I'm going off on a bit of a tangent here, but there's an emerging theme in cellular and molecular biology about liquid-liquid phase transitions. Have you heard about that at all? No. So it's really fascinating. It's really fascinating. It's the idea that there's a kind of a rediscovery of a mechanism to create compartments in the cell, which is based on the physical principle of liquid-liquid phase separation. Like when you mix water and oil, the oil forms droplets because they aggregate and they're hydrophobic. So that's the most classic one, but there are a lot of these mechanisms. Now, reason why I bring this up is because in liquid-liquid phase transitions, there's no molecular order. The proteins or the molecules can interact with each other preferentially compared to interaction with the cytosol. And that's how they aggregate and form these condensates. But there's no order, there's no structure. Here, it's completely encoded in the structure of the triskelion, right? So this structure exists, basically. Okay, let's move on. So no one answered my question about the globular domains. That's for specificity, right? There are two goals that need to be met. One of them is to shape the vesicle, and the other one is to recruit cargo and package them into the vesicle. So you need some specific interaction with the inside. Now, by analogy with what I told you for the ER to cis Golgi transport, here too, everything starts with an off GTP protein, which helps trigger the assembly of the coat. And eventually you need to recruit these cargoes. So if they are transmembrane cargoes, they can, well, sorry. I built everything in the slide. So let's just follow the slides. So the clathrin is just the outside. And here this AP complex, which I wanted to emphasize, is the part that's directly in contact with the cargo. So there's one key advantage to this organization, right? So cargoes, you have either an integral cargo protein, so that's the transmembrane protein you need to transport, and the signal can be directly on that protein because it's exposed on the cytosolic side. If you have a soluble cargo, you need a receptor for one simple reason, which is the coat assembles on the cytosolic side. So this is the membrane of the vesicle, right? But the cargo is inside. So there's no way the AP complex or the coat in general can see that soluble cargo. You always need a receptor, okay? Because the signal has to be facing the cytosolic side. That's where the coat assembles. There's no exception to that. Now, an interesting aspect of the clathrin type coat is, unlike COP1 and COP2, which were directly themselves both forming the vesicle, so providing the structure, as well as interacting with the cargo. Here, the system is modular, if you will. You have clathrin, essentially providing the structure and interacting with AP complexes, but it's always the same clathrin. And there's just a couple AP complexes. But they are the AP complex proteins which are the ones to directly interact with the cargo. So by layering it in this way, you actually create a so-called modular system, right? So instead of having one block for a code, you have two different components. So it creates more viability. Let me illustrate that, right? So the point I'm trying to make here is, so you have different kinds of transport which involve the clathrin code. And I started mentioning that, the trans-Golgi to endosome, and there are two different kinds, plasma membrane to endosome, and in rarer cases, we're not going to discuss much, Golgi to lysosome, and so on. So Golgi to lysosome would basically bypass the trans-Golgi network, right? But don't worry about this. The point I'm trying to emphasize here is that it's the same clathrin that's responsible for making all of these vesicles, but the AP complex can vary. You have AP1, AP2, or this GGA. And so this is what provides diversity, although you're still using always the same clathrin. Okay? And as a matter of fact, because the code proteins here, the AP complexes are not the same, they can recognize different kinds of sorting signals. Okay? So this one, and I don't need you to remember these exact sorting signals, of course. But just to be familiar with the concept that there's a specificity encoded here which determines which kind of code is assembled and which kind of transport. So you can already imagine that it's also going to determine the kind of RAB protein will be incorporated. RAB and V snails, right? Because they are the ones that will encode not the specificity of packaging, but the specificity of fusing with the target membrane. Okay? All right, so let's look into a few more molecular details. So one component, I did not really mention much previously, is this big protein called dynamin, mostly because it's really important for clathrin coated vesicles. So this image is one of those electron microscopy images that has a few components but is very meaningful. Okay? So because what you're seeing, right, so one of those where if I were to show it to you in an exam, for example, you should be able to explain what's going on and what it means in terms of the basic processes we're interested in. Okay, so here what you have is a membrane, plasma membrane, for example. And you see it's formed this pit, this vesicle. It's trying to form this vesicle. But it has this very long neck, which is unusual for a vesicle pinching off a membrane. And it shows this way because these cells have been treated with a GTP gamma S, which is basically a non-hydrolyzable form of GTP. So in other words, all the GTPase or GTP binding proteins that would normally hydrolyze GTP to GDP plus inorganic phosphate can bind this analog but won't be able to hydrolyze it. So in a way, they will get stuck. If they need to hydrolyze GTP to change conformation, for example, they will get stuck in the GTP bound form. And that's the case of dynamin. Now, just to confuse you a little bit, dynamin is not a small GTPase that is only active when GTP bound. No. Dynamin is a molecular motor that uses GTP to exert force, okay, converting chemical energy in GTP into actual physical force, okay? And the force we're talking about here is constriction. So what you're seeing, and the constriction that helps pinch off the neck and free the vesicle from the membrane. So what happens in this image is that you have an accumulation of dynamin here marked by these anti-dynamin antibody coupled with gold particles, right? Gold particles such that they can be visible, they can be seen in a transmission electron micrograph like this one. Okay? So, oops. And so, just before I show you the kind of model, here, because the dynamine cannot hydrolyze GTP, it keeps piling up. But it never closes, it can never constrict the neck and pinch off the vesicle from the membrane. Right? So that's an experimental condition, but it reflects the function of dynamine in endocytosis, so in clathrin-coated vesicle formation, which is to polymerize at the neck here. And once there's enough of these polymers, hydrolyze GTP and constrict, right? And so in the previous slide, what happens is, because the dynamine cannot hydrolyze GTP, two things happen. There's no constriction, right? So the neck stays put, but also polymerization of dynamine continues in excess. And so that's why you have a very long neck. Okay? You can call it the giraffe phenotype if that helps you remember it or something like that. So anyway, so the idea is that once dynamine has helped pinch off that membrane, of course, right, in the first place, the neck and the vesicle starts forming because the clathrin coat has assembled, shaping the vesicle and recruiting these cargoes, and eventually the vesicle breaks off. And then, just like what we saw for the ER to cis-Golgi transport, because there's an off GTP as part of this newly formed vesicle, the formation of the vesicle acts almost like a gap. GTPase activating protein helps off hydrolyze GTP. Now it's GDP bound, and therefore the vesicle sheds the coat. Okay, same principle as what we saw for SAW1. And what's important, right? So that's this first step. HOF1. The complete clathrin coat helps activate ORF1, ORF, the GTPase activity of ORF. ORF becomes GDP bound, detaches from the membrane, that causes the depolymerization of the clathrin coat. So both clathrin monomers or triskelions, as well as the AP complex monomers and so on. Just deassembly. And then it's interact with the chaperone in the cytosol and comes back to be used in new vesicles, right? So again, I mean, you could remember it in thinking it's more economical to recycle these monomers because these clathrins are really big proteins. So if you had to synthesize them de novo every time, it would be extremely expensive for the cell. Okay, so that's what I'm going to say about the formation of clathrin-coated vesicles. And now we're going to switch gears and talk about addressing of soluble cargoes to the lysosome. So one first example of how proteins are being shipped to the lysosome. Before I go in, right, so that's what we're going to focus on. So before I go in, can you take a guess as to what kind of proteins, soluble proteins, so in the lumen, would need to be shipped specifically to the lysosome? What does the lysosome do? Digest, right? Chews things up. Like, what kind of molecule will it digest?
Enzymes.
So the enzymes will be the digestive ones.
Yeah.
That's right. So here we go. We need to ship enzymes to the lysosome. Okay? And those will be really powerful enzymes, right? They can take a protein. There are non-specific lipases, proteases, glycosidases that can basically chew up any macromolecule that gets into the lysosome, regardless of specificity. Now these enzymes, because they are in the lumen, where do they come from? Where are they synthesized? In the ER. Powerful digestive enzymes in the ER doesn't sound like a good idea. And yet, that's where they come from. Okay? So the reason why I played this little game with you guys is to make you think about a conundrum here, which is you're going to have very powerful enzymes capable of digesting proteins and lipids and so on. But you want them active only in the lysosome. And yet, they have to be formed and placed in the secretory pathway at the ER. There's no way around that, through co-translation or translocation. Okay? So that's what we're going to talk about now. So one of the first things that, as you know, happens to proteins when they enter the ER is many of them are glycosylated. Right? The specificity is very low, so as soon as there's this asparagine and any amino acid and a threonine or a serine, it's very likely there's going to be a glucidic residue added. And this is used for quality control and licensing proteins to be exported from the ER to the Golgi through packaging in vesicles. Again, I'm repeating myself. Okay? Now, so this is the step we're talking about. And like we saw last time, for most proteins, there's kind of a default series of modifications of these glucidic residues. Well, it turns out lysosomal enzymes escape that. And they escape that in this way I'm about to tell you. So first off, like all proteins that need to be addressed to some specific place in the cell, they have a signal, right? So they carry sequences or a structure in them that's telling the cell this is a lysosomal enzyme or lysosomal protein, okay? So this N-acetylglucosamine phosphotransferase can now recognize that. And what it does with its catalytic site, depicted here, is it's phosphorylating the residues here on the mannose 6, right? That's why this is called mannose 6 phosphorylation, okay? Very intuitive. So, and that's it. Well, that's almost it. Right? So this is the key first step. And once those residues, those glucidic tags are phosphorylated on mannose 6, they can no longer be modified and recognized by the default modification pathway in the Golgi. Right? So all of this happens as soon as the proteins enter the cis Golgi. This is an activity that's specific for the cis Golgi. Right, so what I'm saying is, so all of this normal modification, which I put there just for reminders, do not happen because these residues are protected from it by the mannose 6. And here, for the aficionados, I just like this word, so I try to place it once in a while. This is a mannose and phosphorylation, mannose 6 phosphorylation means you add a phosphate group on this particular methyl group. And so one of the things we're going to have to think about is how this now will help with, okay, actually I want you to think about that. What else does that do you think? This modification that prevents the cell from further modifying the glucidic residues. Remember, there's one thing we don't want these proteins to do. That's right. So what do you think this tag does in addition to preventing further modification of the glucidic residues? It deactivates them, right? So exactly right. So it helps keep those enzymes inactive or less active. Okay? So in this way, you not only do you protect, it's beautiful, because not only do you protect the enzyme from being modified, but you protect the ER and the Golgi from the activity of the enzyme in one simple step. Okay, one simple covalent modification. Okay, the third beautiful aspect of this modification, it also serves as a specific tagging system, right? Because remember, we need to send these proteins to the lysosome. So with all the knowledge you've now gathered, you have an idea of how this can happen. It happens in the trans-Golgi network, So that's what's depicted here. And here, you know, I could stop, take a break, and let you guys tell me what's going on here. It's a soluble cargo. So what does it need? A receptor, a transmembrane receptor. The M6P or mannose 6-phosphate receptor here. And where's that receptor? In the transgolgi, right? And so how is the system now? Sorry, I tried to build some drama in here. How is the system going to ship these proteins to the lysosome? By packaging them into vesicles. What kind of vesicles? The very same I just told you about earlier in this lecture, clathrin-coated vesicles with AP complexes. Okay? And it's triggered by an ORF GTP, but now the packaged enzymes or these lysosomal enzymes temporarily inactivated by the presence of the mannose 6-phosphate tag, which is itself recognized by the receptor to help bring the enzyme into those vesicles, and now they are en route to where? The lysosome. The first step really actually is the late endosome. So, right, so let's not forget the step where the vesicle has to shed its coat. That's upon GTP hydrolysis by ORF. Now you have a vesicle ready to fuse with the late endosome, and this is where things happen, right? So here you have, so this cartoon is kind of interesting because it represents two essential things. Well, three. One of them is now the vesicle fuses with a compartment which has a lower pH, right? pH 5 to 5.5. That's about 100 times less than pH 7, okay? You all see that? Because pH is a log scale, log 10. And this lowering of the pH does everything you want the system to do, which is deassemble the mannose 6-phosphate tagged enzyme from the receptor, because what's going to happen to the receptor? Recycling, right? And that's the same cytosolic signal here, so it's using essentially the same AP complex in clathrin. Yes?
What is the endosome? Is it another?
So the endosome is, so this term in some cases is a little bit unfortunate. So it's a compartment which essentially is a platform that receives the stuff that's being endocytosed. But it's just that this is a compartment where not only the endocytosed material goes, but also what comes from the transgolgi and is destined to the lysosome. Okay. It's a sorting compartment. Okay, so the last point I wanted to make is depicted here is the lower pH and the activity of phosphatases also helps remove the phosphate. So now what do you think happens when you remove the phosphate? You activate the enzyme, right? So so all of these steps contribute to everything you need to do about these lysosomal enzymes. Inactivate them, protect the system, tag them, send them to the lysosome, and then license them to now become active, but specifically in the late endosome / lysosome compartments. There's quite a bit of exchange between these two compartments. Okay. Just as an aside, because there was a biomedically important aside, which is this system also helps recover misdirected receptors. So they're not really misdirected, so let me just show this. So sometimes you also have a fraction of the receptor on the outside. And actually this can be used if there is mannose 6-phosphate tagged lysosomal enzymes on the outside. This can now come back through to the late endosome and the lysosome, but through an endocytic pathway, which also uses AP complexes, and clathrins. So the reason why I'm showing you this, one reason, is because, well, if for some reason, and there's always a fraction of error in these systems, these lysosomal enzymes are, instead of being directly shipped to the lysosome, they are being secreted, they can be retrieved. And there's a nice example of a disease called eye cell disease, where people have a defect in the trafficking of the lysosomal enzymes. And basically, if you take cells from these patients, so they can't really traffic the enzymes properly. They lack the ability to make the mannose-6-phosphate tag. But if you provide mannose-6-phosphate tagged enzymes to the outside of this cell in the culture, they will be able to endocytose it, and that will rescue the lysosomal function. Okay, so indeed, endocytosis, let's start talking about that, right? Yes?
Are those patients alive?
Yeah, I think so, I hope. I mean, such diseases are not always fully penetrant, so but I don't know much about the medical aspect of it, yeah. Yes?
If you can't phosphorylate the mannose-6, doesn't that leave the enzyme active in the
Yeah, that's a good question, and there may be more problems in this particular disease than that. Yeah. I knew I was opening a can of worm with that comment. Anyway, so let's transition to yet another topic, related topic, and focus more on actual endocytosis now. So indeed, endocytosis, depicted here, is one of the main entry route for material from outside the cell into the cell, usually for compounds that are too big to be transported through the plasma membrane, like you'll see ions or small molecules like glucose. Actually, you may have heard about glucose transporter from Carlos. So endocytosis is kind of intermediate size. You have cells like macrophages specialize in things like phagocytosis. Now, this is for really big elements, such as a bacterium, where you have now the cell is actually protruding to engulf the material. And then you have, and we're going to talk about that not today, but in a future lecture, this fascinating process of autophagy, whereby the cell remodels its composition in terms of organelles. So the cell is capable of digesting its own mitochondria, its own different organelles, and recycling the component. But all of these routes eventually lead to the lysosome. And what I want to emphasize here, so you were asking me about the late endosome. So strictly speaking, endosome comes from what's been endocytosed. And it's just before it fuses with these vesicles that have active, or that are about to have active lysosomal enzymes. So and the combination of both a primary lysosome, almost like a naive lysosome that's never seen any material to digest. So those guys have to fuse with either of these other vesicles to make the secondary lysosome where the digestion and recycling happens. So endocytosis. I've showed you this picture before. I show it again to drive it home, and because it's a really impressive picture. It's as if you were underneath the plasma membrane taking a snapshot and seeing all these clathrin pits. So at this stage, they are called pits because they are not, it's basically catching the vesicles as they are being formed, right? So the material, the plasma membrane is forming a pit, literally, getting internal, and that's driven in part by polymerization of these clathrins, right? So I've just showed you this slide before. This is followed by formation of the vesicle, polymerization of the recruitment of the cargos, breaking the neck with dynamine. So what I haven't told you much about, so I've already commented perhaps ad nauseam about you understand that? That's Latin. So we're forming the vesicle. We've seen that. Okay? ARF. Okay, let's do it another time. ARF comes, GTP bound, recruits the code polymerizes, helps forming the vesicle pinches up. The stuff I haven't told you much about is this essential step of what I did a little bit, recruiting the cargo, right? Never forget, it's pointless to assemble a vesicle if you don't load it with some cargo. And so, what kind of cargo? So that we're going to go through a couple examples here. So a classic cargo that's been studied for endocytosis is these low-density lipoproteins here. So this is the main form of transport of cholesterol in the organism. And of course, cholesterol is essential. You've seen this with Carlos, right? It's an essential component of the membranes, especially plasma membranes. And but it's itself mostly insoluble. I mean, slightly amphipathic because of the hydroxyl group, which in some cases can be esterified, in which case the molecule is completely hydrophobic, okay? And that's why in this particle, the inside is filled with esterified cholesterol. The outside, because it's facing an aqueous compartment, has to be amphipathic, right? Lecture 1, I guess, okay? So there you have, it's like a normal hemimembrane with phospholipids, right, these guys, as well as a whole bunch of cholesterol, but now unesterified such that it has this hydrophilic hydroxyl group, OH, just one, okay? Now, so there's a polar surface, and that's how this LDL can carry a lot of hydrophobic cholesterol, but itself be soluble. Now, it's too big to cross membranes spontaneously. Okay? So those particles need to be endocytosed by cells. But there's very little specificity in terms of recognizing the phospholipids or the cholesterol, right? If you look at this on the outside, well, it's just another membrane. So there's a tag, if you will, that's now a protein, and that's this apolipoprotein B, which forms kind of a belt around the LDL. So now that conveys some specificity. That's a tag that's telling the cells, oh, here is an LDL, not just another membrane. Okay, so now using apolipoprotein B, the cells can recognize LDLs, and they have a handle, if you will, to internalize it through endocytosis. Okay? So let's look into that. So this is, I'm going to show you an experiment that's kind of a classic old experiment, but that's a good one to really illustrate the dynamics of the process. So you start with cells like human fibroblasts, and you keep them very unhappy at 4 degrees, mostly to not kill them, but avoid too much activity, right? It's like, and you load them with this so-called LDL ferritin. So that's something you built in vitro. So there's LDL, and ferritin is just a way, we're going to see some ferritin action after that, but it's just a way to have iron atoms in the system, which are going to be easy to see with the electron microscope, okay, because they are very electron dense. It's just a gizmo to be able to monitor the trafficking of these things. Right, so initially, because all of these cells, they have a receptor for the LDL on the membrane. So the first step is to basically load the membrane with this particle, which now can be seen with the electron microscope. Okay? And the beauty of being at 4 degrees is that nothing else happens. Right? The cell is not really active. So to start the activity, you move it back to higher temperature. Now it can start endocytosing this LDL, and you're going to come in at successive time points to collect cells and do electron microscopy. What you're going to be on the lookout for are these these black dots that come from the ferritin, the iron in the ferritin, that will report on the presence of the LDLs. Okay, so let's just go through a couple images. You see it does make some sense. So very early on, the features that are seen, so that's the plasma membrane. and you see these pits, you see electron-dense material at the base of the pit. That's the clathrin coat. Okay? And then in the pit, but on the outside with regards to the membrane, this is where you have the particles. Well, it's not so easy to see, but you have some of them here and a couple here. This is the LDL ferritin. So it works. You get the stuff you want to endocytose to be where there is a clathrin-coated pit. And then if you look at successive time points, you basically have the whole process. Right? So later on, now you have the ferritin particles internalized, and here you still have this neck. So what do you think is going on here? Dynamin, the constriction, okay? And of course, it's not over, so the electron-dense material is still here. That's still the clathrin-coated pit, okay? And so on and so forth. So formation of the pit, come on. Here, with binding of the LDLs and recruitment of the pit, of the clathrin coats, formation of the pit, formation of the vesicles, pinching of the neck with the LDL ferritin particles inside, then eventually complete pinching of the vesicles off from the membrane with the LDL ferritin inside. There's still a coat. Eventually that coat has to be shed, and later on these vesicles fuse with the endosome, and now you see it's much more diffuse, it's bigger, it's not so easy to see, but presumably there's no more any coat here, right? So that's when the digestion starts, okay? And presumably, so what do you think causes this more dense material? Not just the particles, but overall it's more dense. We've seen something like that before. pH 5, proteins aggregate. Try this tonight. Take some milk, fresh, whole milk, organic, grass-fed, and some lemon. That's gross, huh? And then
Yeah, make fun of them, right?
yeah, well, I don't know how you call that, but you're going to steer coagulation, right? So that's just an illustration that when you lower the pH of a solution of proteins, they tend to aggregate and coagulate, right? So you can say, oh yeah, and then next time you will have this image of the electron microscopy. Anyway, so these kinds of techniques can be used to study the dynamics of the whole process, and it's really fast. Within half an hour or less, the binding goes from the maximum binding at the beginning, and then most of these features disappear because now you have features of internalization, which also wind up disappearing as most of the particles are found in these endosomes, right? So this is very similar to the kind of plot I've showed you for the secretory pathway, except to study the dynamics of another but related process. Now, let's take a closer look at what goes on really at the interaction between the LDL and the receptor. It's an essential one, right? And like I told you, there are, on the ligand side, there are these different components. That's the LDL particle. There are the esters, the cholesterol, and phospholipids, but it's mostly the ApoB protein is the signal. And sure enough, this is the part that interacts with the ligand binding arm of the receptor. So now that's the receptor. So I want to emphasize a few features of the receptor that makes this work. The first one is the ligand binding arm. We're going to talk about the propeller domain in a second, or a couple seconds. And the other one, very important, is this NPXY sorting signal. What's up with that? Come on. What does that do? Where's the NPXY? In the cytosol. What does that do? That's right. It helps recruit the coat. That's the signal. Because this is a soluble particle, the coating machinery does not see it. It needs the receptor, it needs the signal in the receptor. Right? Are you connecting the dots? Not just the electron dense dots, but the different dots. Okay, so that's essential, right? And that explains at the molecular level how you can connect the LDL, the soluble cargo, with the recruitment of the coating machinery via the receptor. And sure enough, for different kinds of receptors, for this so-called receptor-mediated endocytosis, you have a specificity for the ligand, but you also can have a specificity for recruiting the coat. So, for example, to send membrane proteins in the trans-Golgi, it's a slightly different signal to send them to the plasma membrane, and so on and so forth. Okay. Now, let's just go through more details about what happens next, right? So the vesicle forms integrating this ligand and the receptor, the coated vesicle forms, sheds the coat. Remember why it sheds the coat, right? Because the GTPase has hydrolyzed GTP. Now it's GDP bound, the coat goes. Okay? Early endosome, the LDL is still bound. Late endosome, it's no longer bound. The key difference, we've seen that before, low pH, more acidic compartment, pH 5, versus at the cell surface, it's pH about 7. Right, so it's another case of the pH 100-fold difference in terms of concentration of protons causes, regulates the affinity for the receptor for the ligand, right? Regulates the binding. So I just want to illustrate how that can work in this particular case, right? So 7 versus 5, how does it work? Well, one first question is, well, first, how do you get pH 5 in the late endosome? How do you increase the concentration of protons so much, about 100-fold compared to the cytosol in the outside of the cell? How? Do you have an idea? A proton pumps, that's in the slide and you have heard about it, but yeah, good one. That's a proton pump. Now, let's just think for a second about the proton pump. It's pumping protons, it's a proton pump, from the cytosol into the late endosome. So is it following, in which way does that go compared to the gradient? How is the gradient of concentration? Not everybody at a time, because I don't understand. It's against the gradient. So is that going to be an endergonic or an exergonic reaction? Endergonic is going to require energy to pump protons against their gradient, uphill. So indeed, and these kinds of enzymes, this so-called V-class proton pump, does that structure ring a bell? Yes. Yeah, it's like the ATP synthase. So it's an ATP synthase working in the opposite direction, essentially. Hydrolyzing ATP, so harnessing the energy of ATP to couple this exergonic reaction with an endergonic reaction, and that's transporting the protons back, or from the cytosol to the late endosome. 100-fold more highly concentrated. Now, that doesn't tell us how this difference in pH regulates the binding of the LDL to the receptor. So that's what we're going to look at now. And so here is a model of the structure with the ligand binding arm and the propeller domain I was showing you. So in the closed configuration, those two come together. And the idea is that closing this opposes the binding of the ligand arm to the ligand, to the LDL ligand. Okay? So what you want is the low pH, high proton concentration, to favor this configuration over the open one. And the simple solution is because protons are positive charges, and at lower pH you have residues like amines that can be NH3 plus instead of NH2. Now those charges, they get here. And it so happens that there are negative charges in the ligand binding arm. So now, because you're at lower pH, you have now strong electrostatic interactions between residues. Whereas at higher pH, those residues here are not positively charged, right? So the interaction they may have with the ligand binding arm is weaker, right? So if now it competes with the LDL, binding to the LDL wins at higher pH, binding to the beta propeller domain wins at lower pH. That's how you regulate this activity. Okay, so what time is it? And we're good. Next, what does the cell do? So so what is done before is taking the LDL and channel it all the way to the late endosome and dissociate the LDLs from the receptor. So importantly, that receptor is now going to be recycled. Okay? And presumably, so the cartoon here do a good job at conveying that those vesicles, they come from the late endosome, they have low pH, so the receptor is in a closed conformation. Once they reach and fuse to the plasma membrane, the receptor faces a higher pH, 7, it opens back up. So it's now basically ready to bind to and help endocytose and other LDLs. Now, these recycling loops, right? The endocytosis and recycling goes really quickly, maybe half an hour, give or take. Those receptors have a half-life of many more hours, and the estimates are that they go through about 700 cycles of endocytosis recycling. Okay, let's move on because I wanted to show you another example. So this recycling, just a quick note, is extremely important. So we saw it for M6P receptor, for this LDL receptor, and I'll probably tell you a bit about recycling of integrins when we discuss cell matrix adhesion. But for now, let's just look briefly, or thoroughly, but at a different kind of endocytosis, just to illustrate the idea that what I'm telling you are kind of general principles that can apply, with specifics in many other cases. So that's the transport of iron. So I thought to keep you awake, because I've been talking for about an hour. So what kind of roles can you think of for iron inside cells? Hemoglobin, that's a good one. That's the first that comes to mind. In every cell?
No, just red blood.
Now in every cell. Some that you've seen with Carlos.
Electron transport?
In the electron transport chain. The iron-sulfur clusters that helps pass electrons around, right? That have their redox potential modulated by the surrounding amino acids. Some of you look at me like, oh, how does he know that? Was he in the class? Well, I used to teach these lectures. That's why. Okay, so iron is very important. The cell needs to internalize it, but it's a tiny ion. There's not much specificity to it unless it's bound to a protein. And so there's a transporter for iron. That's the so-called phosphotransferrin. So it comes in the blood and on the outside of cells in this form of phosphotransferrin. And here too, pH has a role to play, and differences in pH between compartments. Right? So just like for LDL, where for LDL, what matters is the cholesterol, but the cholesterol can't be recognized directly, so there's a protein to tell there's cholesterol, the apoprotein. Here there's transferrin. There's a receptor for transferrin because it's a soluble one. And you need a receptor with a cytoplasmic tag to recruit the coat. All the same principles. Recruit the coat, form the vesicle, shed the coat, fuse to the endosome. Now, the changes in pH, here there's something slightly different, which is because it's iron, and its own charges can be modulated by pH. The lower pH causes this transformation from Fa3 plus to 2 plus. It's just the redox potential of iron that does that. And now this is enough to dissociate the iron atom from the transferrin endoreceptor. Right? So here what's decoupled is the iron itself goes out. And then it's so small that there's a transporter to take it through the membrane. On this side, the transferrin is now called apotransferrin because there's no more iron, but it's still bound to the receptor. And it's still bound to the receptor because it doesn't matter in this particular case if there's a lower pH. Or, I should say, the apotransferrin without iron binds to the receptor just fine at lower pH. And then the whole system gets recycled and now it's slightly higher pH. So this is the subtlety of the system. The apotransferrin dissociates from the receptor at higher pH. So you might be, it's confusing, right? Because here it associates, except that here it's bound to iron, not here. So you have all of these affinities that are modulated by the exact situation, which is presence of iron or not, lower or higher pH. But by and large, the whole principle is exactly the same. Okay? You modulate interaction to endocytose or scavenge the compound, take it to the late endosome, recover it, and then recycle the transporting and receptor machinery. All right, so all of these addressing, this is in a way a bit of a reminder, but in all of this transport, right, yet again, vesicles have to go to the right place. And we saw it a few times already. It involves Rab molecules, it involves V snares. So here I just want to illustrate the idea that we have, and I'm going to probably talk about those again when we discuss integrins. So Rab5 usually helps with the early endosome. So when vesicles are first endocytosed, they know to go to the early endosomal compartment because they have a Rab5 tag. Right? So the Rab5 tag, you could imagine, is actually added to the vesicle upon endocytosis. Right? I didn't illustrate that in the previous slides, but that's what happened. And then from these early endosomes, some vesicles are targeted to the lysosome, go down this lysosomal pathway, and they use the Rab7 tag, whereas Rab4 and 11, they actually contribute to recycling, right? Because, in a way, the reason why I put this slide here is because it's a conundrum or it's a question from the previous slide. In the same compartment, the late endosome, how come some compounds are destined to be recycled to the plasma membrane, other compounds are destined to stay in the lysosome or fuse with the lysosome and get degraded? It all has to do with the packaging of the vesicles, and in this particular case with the Rab tag. Okay, almost done. So one thing to keep in mind is reaching the late endosome is not the end of the journey for these endocytosed components. And one of the ultimate goals, indeed, like we already saw, is to recover, let's say in the case of LDL, is to recover the cholesterol molecules, the fatty acids, and so on. So, in other words, whatever is being endocytosed has to be broken down into simpler components. What does that? Breaking down? The catabolism. Okay, so you guys are really going for the high road here. Specifically, what kind of proteins do that? Yes, have we heard about them today? Yes. The lysosomal enzymes, right? So this is where the lecture has different parts converging, right? Because the lysosomal enzymes we talked about, mannose 6-phosphate, yada yada. Now they have to fuse with whatever is being endocytosed if we are to digest this. Okay, so indeed this all happens, right? So these enzymes are trafficked here. I showed you that earlier today. So the whole idea is that now you have in the late endosome or early lysosome compartments, this is where you have to merge what comes from the secretory pathway and contains the enzymes and what comes from the endocytic pathway. Okay, and yet, and here I'm just going to give you a teaser and a harbinger of things to come in future lectures. You still have to sort things out because there are enzymes and lysosomal membrane proteins that you do not want to digest. But there are also enzymes membrane proteins from the endosomes. Some you want to digest, some you want to recycle. So we're going to have to see and understand how this is distinguished. And I'm just going to finish by showing you this image of things to come. So those are all views of different lysosomes. And you see it gets very complex. Right? So in some cases, this is actually not a lysosome. It's a lysosome that comes from an autophagosome. So here you have organelles, a mitochondrion, and a peroxisome inside another compartment. Those are fated to be digested. And here you have a late endosome, and what do you see here? This is an autophagosome. More vesicles. So this is called a multivesicular body, and we're going to see in greater details how one way lysosomes sort out what to digest, what to not digest, is to create intralysosomal vesicles or not to distinguish between digestion and either recycling or lysosomal membrane proteins. And I'm going to stop here.